Hello everybody. Today we will be discussing animal diversity. Animals are heterotrophs and they ingest the food and later digest it using enzymes. Animals are also multicellular but have no support structures like plant cells do with their cell walls. Instead, there are special proteins. Animals also have two kinds of esoteric cells, muscle and nerve cells. They make the muscle tissue and allow the nerve impulses to be conducted throughout the body. This is a distinct feature of animals. Most animals reproduce sexually, and the zygote goes through a certain process during the embryonic development. The zygote first undergoes cleavage, a process of successive cellular divisions without any growth. Then it turns into blastula, a stage where it is multicellular. Inside the blastula, it is hollow. Then the embryo goes through gastrulation, a process where one end folds inward and fills the hollow space known as blastocell. This makes layers. The outer layer is called ectoderm and the inner layer is called endoderm. Sometimes there's a layer in between, which is called mesoderm, and we'll get to that soon. The pouch which is formed is called archenteron, and the thing itself at this stage is known as gastrula. The bottom hole, as you can see there, is called blastopore. A lot of animals, including humans, grow directly into adults, but a lot of other animals also go through a larval stage. A larva is a young animal which does not resemble its adult version and is sexually immature. After undergoing metamorphosis, the larva resembles its adult version but is still sexually immature. Another little side fact is about genetics right now. So we know that genetics, they control our traits, right? The expression of our traits. But there are also genes which control other genes, and they're known as regulatory genes. Regulatory genes contain sets of DNA which are common to all regulatory genes, and they're known as homeoboxes. Animals' homeoboxes are called the Hox genes. Just a side fact. Now, another little side fact again, just the history of animals and life. So, during the Ediacaran biota, it's about a billion to 542 million years ago. We had the first life appear around that time. And then from that to 251 million years ago, sometime during that time, the Cambrian explosion happened. And as most of you know, that's when all of a sudden the diversity in life just boomed. And then soon, a little while later, the animals started moving to the land as well. Another way to classify animals is through their symmetry. Animals have two kinds of symmetry, a bilateral and a radial one. And some have no symmetry, like this sponge right here. So bilateral symmetry is simple. You have a left and a right. It's what we know as symmetry, what we've learned in math class, right? It's like a mirror image. But then there is also the radial symmetry. This one's weird because it's like a sphere, right? It's symmetrical from every side. Yeah, that's what it is pretty much. It's like a circle, it's symmetrical from every side. There are no left or right, like everything is left or right. Bilaterally symmetrical animals have an anterior, the front side, the posterior, which is the back side, the dorsal side, which is the top, and then the ventral side, which is the bottom side. And the mouth and the sensory parts are usually on the anterior side. And it's simple to remember. Everything, our senses, most of our senses are in on our face, on the front, because we're also bilaterally symmetrical animals. Going back to tissues, some animals have two and some have three. So they all have ectoderm, which is the outside layer, and the endoderm, which is the inside layer. The ones which have three layers also have mesoderm, the middle layer. The ones which have two are called diploblasts. The ones which have three are called triploblasts. And here's a neat little image showing their cross section. So as you can see, diploblasts, they do not have the mesoderm, and there could be uh, like a jelly-like substance in between the two layers. And then, of course, the triploblasts, it's got the mesoderm, which you can see is represented by the really dark color. Now, triploblasts often contain a body cavity, which is known as the coelom. The true coeloms are only in the mesoderm, and they only connect at the dorsal and ventral areas. Everywhere else around, there is nothing. Usually, it's either just air, or it's like a jelly-like substance. Anyways, animals possessing such trait are called chelomates. There are also pseudochelomates, and they have a cavity, but the cavity is extended to the endoderm. It's not only in the mesoderm. And then you have the achelomates, and they have no cavities at all. You can see all of this in the picture. You might ask, what's the benefit of having a coelom? Well, the fluid inside it cushions the organs, then it also supports the muscle, and it allows the organs to function properly by allowing substances to travel in different directions and different ways. Animals can be also categorized as protostomes or deuterostomes. They differ in the cleavage, the coelom formation, and what happens to blastophore at the end. Protostomes begin their development with a spiral and determinate cleavage. This means their cells have been determined for future in terms of what they will become and what will happen to them. But deuterostomes begin their development and have radial and indeterminate cleavage. This means that every cell can become whatever it can. It's not determined from beginning, and it can even become its own embryo. This is confusing, but think about twins. That's why having twins is possible. Okay, also in protostome development, the coelom forms from the gap in the mesoderm. Simple. But in deuterostome, the coeloms form from the pocketing of the archenteron. And then finally, the blastophore. In protostomes, the blastophore becomes the mouth. And then a second opening appears, which becomes the anus. In deuterostomes, the blastophore becomes the anus, and the second opening becomes the mouth. And this is how we distinguish between protostomes and deuterostomes. Phylogeny is the development and diversification of life. But there are two different life trees. One is based upon morphology of animals, and the other one is based on the molecular data gathered. Although they're different, all the scientists agree on five things: that there is a common ancestor to all the animals. Sponges are at the very base of the tree, as far as our knowledge allows us to go back. And eumetazoans have tissues, and that's where the tissues started in human animal bodies. And most animals have a bilateral symmetry, and they have three tissues. And chordates and a lot of other phyla, which are the categories of animals, are deuterostomes. So yeah, guys, this is it. And if you have any questions, please message me on Facebook or email, leave comments, whatever. I've spent a lot of time with this topic, so I should be able to answer all, if not most of your questions. I mean, sorry, most, if not all of your questions. And I just wanted to finish off by recommending this book. Again, I've recommended this over this entire academic year, whenever I could. I'm just doing it again. If you really like, like evolution, history of life, and humanity, global issues, all that kind of stuff, you will love this book. It has become my favorite book after the Harry Potter series, which are the only reason is number one is because of sentimental value. And yes, please, during the summer, read this. This is a great book. You will not regret. Trust me. And I just want to say thank you for listening to this, watching this. I mean, yeah, also, of course, you have to, but still. And yeah, make sure to send me any questions if you have them. Thank you very much. And all the sources have been cited in the next slide, so no plagiarism.